ch14 clinical development strategy for cancer treatment. So conventionally, they use chemotherapy using different chemicals and drugs to target generally target cancers, um, but and they also use radiation therapy just to kill damage the DNA of the cancer cells to induce apoptosis and immunotherapies. And they kind of generally just hits everyone like this. They treat them all like same way, which isn't the best way to deal with cancers. So they're developing targeted therapy for cancer, more like a precision medicine. So, for example, there's a target DNA repair pathways. There's ThinkAPARP, which stands for polyadenosine diphosphate ribose polymerase. What they do is there's an enzyme that repairs DNA and keeps the cells alive, and which is good thing for normal cells, but for cancer cells, it can helps cancer cells keep living, like stay survive. So they develop a PARP inhibitor along with BRCA1 and 2. Uh, they're also a DNA repair enzyme. So by uh, with those inhibitors, they develop a synthetic lethality, which what it really does is just they're damaging all the cells, including cancer and healthy cells, and cancer will die off more quickly than healthy cells because they already have damaged DNA. Observation, questioning, experiment, analyzing leads to new knowledge. So this just shows the BRCA2. Um, it's, they, since the discovery of BRCA2, it took 25 years for it to become a cancer treatment. It takes a long time. There's ecDNA, which is extra chromosomal DNA pieces that's floating around in the chromosomes outside of chromosome. Um, it also took a long time. So here there's a naive cells, and then there's a drug resistant cells and drug removes cancer cells, where red represent the ecDNA, which is EGFR, which is something that helps cancer to grow. Um, so for the naive and drug removed cancer cells, there's a lot of the ecDNA, but when there's a drug, when it's treated with drug, like they kind of get combined into the chromosome to and interfere with drug acting on ecDNA. So they used to use microarray um, for like for testing all the different type of genes that's in the cancer cells, which could be like hundreds and thousands of testing. But now they use RNA sequencing, which takes a lot less time. And they use microarrays to see what kind of to gene profiling and pick the right best of treatment based on that. Um, Biomarkers, just like a features or type of molecule that can kind of shows what kind of disease or underlying things they have. So prostate cancer biomarkers in urine helps avoid unnecessary biopsies. Similar to colon cancers can be detected in poop samples. Circulating tumor cells could be also used to detect cancers in blood. CRISPR-Cas9, known as genetic scissors, is a thing that already exists in nature, which is derived from prokaryotic adaptive immune system, and it's designed to destroy the DNA insertion of viruses. When the foreign RNA was detected, and what it does is it's composed of the single um, the single guided RNA, and this RNA sequence leads to a place in DNA that matches the sequence, and the scissor kind of cuts that targeted DNA. Um, and, one of the like side effect could be that if the RNA sequence is if there's identical RNA sequence in the DNA, then it can also cut those unintended matching sequence of DNA. unintentionally and cause unwanted side effect. They're using this CRISPR-Cas9 for editing PD-1 in T cells, which is this thing on T cells. So basically, tumor and T cell interact with PD-1, and you basically and helps tumor survives. And by we can modify it so the T cell doesn't develop as much PD-1 and help cancer tumor cells to die. CAR T cells, and so these are immunotherapy examples. CAR T stand for chimeric antigen receptors, basically a combination of the antibody and then the T cell receptor. That so the T cell can directly bind to the antigen of our choices by designing an antibody. Cancer nanotech, anything that's deal with nanoscale stuff. So it could be gold nanoparticles, liposome, which is just a fat, fat vesicle that helps guide stuff to right places and such. Imaging technique, CT uses X-ray, but X-ray causes with the risk of cancer. PT, positron emission topography, and they uses the Warburg effect, which just saying the cancer loves sugar, so they have more sugar in there. So we can use a glucose analog radio tractor, which is just sugar with more radioactive stuff in it, and we can use PT to detect the presence of radioactiveness in different parts of the body, and hence detects cancer that way. Optical imaging, which is just similar thing that we do in the microscope, just in the body, MRI, radio frequency, and they use it on people, but can't have metal in their body. And ultrasound, it works for superficial stuff, but can deal with the deep stuff. Drug development stages. So they first have to set the molecular target on what they're targeting for, different pathways or just process. They optimize it, and then they do preclinical study, clinical studies, and get approval from FDA. In the process, they use like animal models such as mice, where they plant a human tumor in the brain of the mice, and they use immunodepressed suppressed mice because they don't reject the human cells. High throughput drug screening. They test a bunch of drugs on different cells, and it could be used to discover the new drugs that works or modify the existing drug. And they test different versions of it and find the enhanced version of the existing drug. Phase one is yada yada yada. So the problem with the clinical trial is the recruiting stage, only the like people, hopeless patients volunteer for it. So it's like inherent bias, and until phase three, there's no control involved. So that's another uh, issue. Phase zero trial often held with less than 20 people for 1 week only, so it's not really enough. And phase four happens after the approval by FDA, where they evaluate long-term risk and benefits. These stuffs are great, but the cytostatic drugs doesn't really fit into this current trial method because it's more of they're designed for the longer survival rather than reduction of cancer tumor. So there's a lot of targeted therapeutics, these targeting different pathways and cancers, but they're more expensive because they're more selective. It doesn't apply to everyone, so less number of patients, so they have to charge up more to make up for the R&D stuff. But it shows that inherited genetics may affect the outcome of the drug. And we're making progress because cancer death rates fell by 27% since 1991, and survival rate increased, and we have immunotherapies and others going on, but it's easier to prevent cancer than cure. That's it.